Hi everyone, this is Aleyna. Today we present exploratory data analysis for lung cancer histological images. Lung tissue classes, we found a source of our lung tissue image from a Kaggle dataset. In our machine learning project, we have three different classes. ACA and CSS are cancerous lung tissues, forms in the organs while CSS forms in sclerotium. In our IDA study, we took smaller health-related IDA and ML studies into consideration. There is our data set and some photos and images of cancerous lungs and cells.
Hello, my name is Furkan Yalçınkaya. This is examination of dataset. You can see the difference between normal ACC and SCC classes.
So what you see the difference?
Difference, as it can be seen, images, cancerous tissue are more purplish than the healthy normal tissue images. And the reason for that is because in order to examine these tissues, they are added blue-purple colored special liquids and set for drying, as a result of which we can visualize that cancerous ones are purple or blue.
But do you think that the color is enough? Is there any difference between in the texture?
For example, SCC.
Can you go back?
More than purple.
Can you go back?
Okay.
Sorry. Go back. What I am suggesting is that you are talking about only the colors of the images. I am saying that, is there any difference in the patterns? For me, it's seen that the small dots are much more dense in AC and SCC classes. In normal classes, I see that many vacant areas. Yeah. So do you know this? Because if it's only colors, then it's a color detection algorithm can help you. But if it's not only the colors, so please pay attention in examination of the images. So now you are very oversimplified the problem, I believe.
Yeah, it's a problem, but we can find a kind of algorithm that you mentioned.
I am mentioning that. I don't believe that it is only color. Do you understand this?
Yes.
But you are only mentioning about the difference in colors. Do you understand this, Furkan?
Yes.
We try.
Can I add something?
Yes,
I'm Önder.
Yes,
so today when we were working with Furkan, he also mentioned that the ones with cancerous ones, he said they are more dense. I mean, he was always asking me about why cancerous ones had more black dots, as you can see in the below images here. And as if you look at this one, usually normal ones have more. For example, let's check them here. The normal ones have red dots, and the other ones with cancer have more black ones. We have visualized.
But Önder, are you sure that these images are taken by the same microscopic device or not?
Yes, they are.
Yes.
Are you sure that they are taken with the same specific?
Yes. We mentioned this in the following slides. And
yes. Because the title is examination of data set, it shouldn't be so simple. Otherwise, the doctors would see that, oh, it's a purple, okay, it is just the ECA or CSCC. Do you think that it is so simple? Only the colors.
According to my researchers, doctors can say just by looking at the image. And it's, as I mentioned, these colors are put on these tissues on purpose because they are set for drying for a few days. And with the colors, this affects the lighting of microscope. So if they are cancerous, they seem to be bluish and purplish.
Do you have the any written report like that? Do you have the
We have written these in the senior project, I mean final report.
Okay.
I'm getting help from my sister who is studying medicine.
Okay.
I read
So
these
mention this as
Okay.
a providing reference to your
Okay.
sources. Okay.
Okay, I read these and took the references.
Okay, I please add it up. Okay.
Okay.
Let's continue.
I want to continue. We look sample numbers for each class, and the following code prints the number of samples for each class by counting the number of images in the folders. Considering the result of the code below, it is noticed that the number for each class is the same. So the samples of the classes of the dataset are balanced. Also, since they are counted using their labels, it also shows that they are all named correctly.
So are these numbers for the training data set, test data set, validation data set, or just the data set?
Just the data set. We have not done our pre-processing and machine learning study yet, because you mentioned that you didn't want any machine learning model.
Yeah, but I am saying that some data sets are coming with the splitted data sets. So nobody splitted this one.
Sorry, can I get the question again?
Sometimes the data sets are just splitted by the publisher.
Yeah, these are separated.
I mean that in the train set, validation set, test set, they are separated and put into different folders, as we discussed in the previous slide. So in the previous presentation of the other group.
Let me show you here.
Directory.
In the directory, as you can see, ACA, normal, and ACC are different folders, and they're all inside the same folder. I mean, there's no training and validation data set.
So just mention about this, okay? In your
okay. Okay, we will add it.
Okay, it is noted.
Now it's my turn. Hello, everyone. Sorry?
Who are you?
Yeah, I was going to introduce myself. Yes. Hello, everyone. This is Önder Ertan. Image sizes and color channels. In the following example of our code, we printed out our dimensions, I mean sizes, and the color channel of our images. We have randomly picked, actually we have chosen the first images from each directory, and we have printed their sizes and the dimensions for each. As you can see, they all have the same pixel sizes, and 3 in this one refers to the different color channels. So they all have red, green, blue in them. We have, of course, made a research about this function, and it's about slicing and indexing. So also all of these images are taken using by the same microscopic device. That's why they all have the same sizes. So now we are going to talk about duplicates. Today we made a research on our images. I used diffpy library to search for any duplicates in the directories. So the library checks each image by rotating them. I would like to
start you at this point, intervene. I am sorry. Önder, it's a good idea to check the duplicates. Thank you for that.
You're welcome. So the function, the library uses a function that takes each image and rotates them and compares them. So in our research today, we found out that only in the normal directory, there were 486 duplicates. So in the previous slides, we mentioned that all of our data sets have had the same amount of images, right? But if you remove these duplicates, we will have an imbalanced data set, we believe, because only in normal one, there are 486 duplicate images. Wait, let me show you in the next one. So as you can see, this function from this, this library we ran it in the normal one, and in 31 minutes, it found out that there were 486 images. Here it shows that this is the original one, and this is the duplicate. So as you can see in this one, wait, so excuse me. So in this one, for example, this one here, this part here, when we rotate it, it's here. This is lung normal 1003. This is lung normal 1492. They are the same images, just rotated 180 degrees, as you can see.
So we can say that somebody also did data augmentation by just turning, rotating the images.
I think they took the images. I mean, I don't think they duplicated them, but they took the images by rotating them or from different sides.
Okay, in machine learning,
yes,
it is called data augmentation technique.
Oh, okay.
In the data augmentation technique for the images, most of the time we rotate the image, we slice the image, we just put the image in another shape and increase the number of the images by that. It's called data augmentation. You are gonna do this in your project, okay? And I think that in that data set, it is done by somebody else.
Okay.
So it's a good idea on that to check these duplicates and find that this data set has some already data augmentation techniques already implemented or applied to these. So it's a very good idea to do this EDA. I will add this EDA to my documentation as well. Check the duplicate. Thank you.
Okay, I have done this with my friends because I thought that this could cause overfitting in the machine learning process, because as you know, overfitting is when computer kind of memorizes the images, and when we add the testing value, testing image, let's say, it just gives us the instant answer, and it usually has high accuracy because it does memorizing, as I mentioned. That's why we added that. Thank you for listening and your time, sir.
Okay, so you didn't provide us, actually you can do, if you go to the, please do not stop the sharing the presentation.
Oh, sorry.
If you go to samples, if you go back to the samples, sample images from each class at the beginning. But there are more. Yeah, in that class, we can see something, right? So you need to provide some statistics about the images.
Yes.
You remember that in the previous presentation, Bilge has already compiled information about the images into a CSV file, right? Height, width, and resolution, anything you can collect from the images. That please report these dimension, averages, mean, maxes per these three classes.
Yes.
You could see that normal class say, for example, I don't know more about the minimum values, these, maximum, these. So you can do this EDA, but now you are missing. So we are just talking that is that only the colors or the some dots or the, I don't know. Do you understand that? You need to work on a little bit more on reporting the statistical information in that images per class.
Okay.
Mean, max, average, or distribution.
Can I just show you something something from our IPYNB file?
Yes, please.
We were using Jupyter Notebook Lab. Okay, so I mistakenly deleted, but all of the images in all folders have the same sizes. I checked them, but last night I slept kind of late, and by mistake I deleted them. I'm so sorry, but they all have the same sizes.
Find the histograms, right? For example, the color values, histograms.
You mean these, right?
Yes, this kind of things per class.
Yeah, but
Maybe three
I will,
classes. You can
I
get
will,
the average of them, and then
yes,
maybe you have some difference between the classes. Do you understand this kind of
yes.
thing?
We have some questions actually. So according to our researchers, this one refers to RGB channel, but when we do a difference between, for example, ACA1 and ACA5, we get this image and it makes nonsense to us because we can't see the labels. So we would like to know if you know what these label means, because all I can see that this goes to 700 something. This can be the sizes. I mean, 768, 768, you know?
In that code, actually, you are just comparing some two images, and they are just getting the subtraction of the pixel values.
Oh,
So
okay.
what you suggest to end up with, so I don't understand the reason of that code.
Um, so I was as you can remember, we referred to we took in consideration some other health related machine learning studies. In these ones, they were usually comparing images of, for example, chest images from pneumonia related
But
issues, you know.
yes,
So I was doing that.
they are okay, I understand. But please pay attention what would they use in, right? Now they are just, if you are just removing one image from the other images, it wouldn't provide you the information, but you can collect the information for the image histograms. Then
Yes,
you can get the average of these histograms per class. Then
yes,
you can report this, and then you compare the results, comparing the with different classes. So the labels, three classes here, three labels here.
yes.
It's that you can tell that the mean values of the colors is that, but in that class the mean value is that, or mean max values. The distribution of the pixel values would be different. I believe that it would be different. Okay, in grayscale, if I were you, if you go up your first slide,
Yes, sorry.
there are many examples right here, this slide. So if you convert all of them to gray scale, for example, then
Yes,
because your model will work on the gray scale most of the time, right?
yeah,
So
okay.
maybe you will show us that even in the gray scale, you have some differences that we notice by our eyes.
Oh, okay.
But now it is missing.
Mhm.
So there could be many other different techniques to apply to your EDA,
Yes,
but helps you to understand how to differentiate these three classes. Do you understand? That's the
yes,
reason why we are conducting an EDA. Do it in many different ways, okay? Think
okay,
about that, because EDA enforce you get familiar with your data set, images, classes, blah, blah. In the end, when you begin to build your model, you will build your model accordingly, according to your insights gained during this activity. So please pay attention and continue, extend your EDA with different techniques. Do you follow
okay.
me?
Yes, yes, sir.
And the other teammates?
Yeah.
Furkan? Aleyna?
Yes.
Okay, so for the moment, it's a good beginning point, but
Yeah.
please think about the other techniques that other groups already presented. And according to my email or the thing that I already shared with you, how to do EDA, as simple ideas, add these simple ideas to your data set EDA as well. Is that okay?
Okay, all right.
All right.
Thank you, sir.
Anyone would like to add something?
So I have a question,
Yes.
actually.
Yes.
So in the previous one, you know, we found out that there were duplicates. Do you think we should remove them?
No, you don't need to remove them, because you will see that at the model pre-processing type or pre-processing phase in machine learning, deep learning, image processing, you will apply data augmentation already.
Okay.
You need to. So pay attention that you can follow two different paths if you like. You can remove the duplicates one, then you apply your pre-processing, I mean data augmentation, rotation, slicing, blah, blah, blah by yourself. Okay. Or you can use this data set and apply these things onto this data set. It's up to you.
Okay, thank you.
But just mention what you do or plan to do in your report.
All right, we will do that.
Don't forget. Okay.